Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Rod Scott, Professor of Neurology and Division Chief of Pediatric Neurology at Nemours Children's Hospital in Delaware. Today, we will be discussing how Rod bridges his research in the clinic to care for children with epilepsy and additional cognitive and behavioral impairments. So welcome, Rod.
Thank you very much. I'm delighted to be here. Looking forward to it.
You have a very challenging position, I believe, working in the clinic with children and then also doing research on the diseases. You once described, I think in a paper, the brain is being very complex adaptive system. What does this mean? And how does this perception help us understand, epilepsy or the other neurodegenerative diseases?
So the idea of complex adaptive systems is really reasonably new compared to the more traditional approaches to understanding brains. So a complex adaptive system is defined as something that is characterized by many interacting parts that are often organized in a hierarchical way that can learn and adapt over time. And this leads to emergent, often unpredictable behaviors. So the thing with these systems is that they are dynamic, they exhibit self-organization, there are characteristics about them. And the statement that's often said about complex adaptive systems is that the whole is greater than the sum of the parts. So there are characteristics that are really important when we're thinking about complex adaptive systems, and these are protean. They are everywhere. They are ecologies, economies, traffic and airline networks, internets, brains. Just about everything is a complex adaptive system. And there are strategies for dealing with complex adaptive systems and understanding complex adaptive systems that haven't really made their way yet into neurology. So there are things that make a brain explicitly a complex adaptive system. And characteristics of all of these systems are things like interconnectedness. There are many parts, there are many individual elements, they talk to each other, they interact with each other. They're adaptable, so inputs into the system changes the way these elements talk to each other, and then the output of the system is different. And the outputs of the system is what we call emergence. And there are no straight lines. So you can't say, because I break gene X, there's a straight line to epilepsy, cognitive impairment, Alzheimer's disease, autism, anything you like. The lines are much more wiggly than that. So there's a non-linearity that arises from feed forward and feedback things happening inside the system. And a critical piece of this is that they self-organize. So an ant colony or murmurations of birds would be good examples of these. So if you watch ants, or if ants are building a termite mound, they build these amazing structures which have purpose. They keep the ants cool. They stop them from dying out in the Australian and African deserts. And these are not because there's some king ant or some king bird that's driving the shapes of murmurations of starlings, for example. It's because how they all locally interact, and those local interactions lead to this emergent thing. So this is critical, because if you want to understand the shapes that the birds are making or the termite mound, you can't take every single ant or every single bird and study them individually and then add them all up and make a termite mound, or add them all up and make a murmuration of birds. And in the context of brain, biology in general, you can't take all of the genes, understand each one individually, add them all up and get a human. There's something extra. There's that additional piece that's emergence. And out of that emergence, well, from that emergence, you see patterns. So that's really what we are interested in studying. We're interested in studying patterns that the brain makes and whether those patterns are altered in disease and how we might be able to recover some of those patterns. It's important that I say at this point that even though you're talking to me, what you're really talking to is a group of people. So we are very much an interdisciplinary set of labs. There's me that brings medicine and some neuroscience, particularly pathophysiology of disease. Amanda Hernan is my colleague who's a basic neuroscientist. So she brings straightforward neuroscience. And Matt Mahoney is a mathematician. And we meet every week. We discuss things. Our labs are all deeply integrated in a way that we're able to think about these ideas in an interdisciplinary way and apply them to neurological disease.
It's so interesting because, my discipline, my background is engineering, and you mentioned some of the other kind of systems, right? Our transportation systems, our, financial systems, and how these systems of systems adapt to changes in kind of the subsystem or the tertiary system. How are you studying, or are you studying those other, areas to try to understand what's happening in the brain? Because, like traffic on a highway in any given day, there's a lot of it flows through the highways. But if something happens on the highway, they adapt, they go down the back roads, and then all of a sudden the highway's clear and everybody gets back on the highway. So how are you, or are you studying other systems?
So we are not explicitly studying the other systems, but what we strongly recognize is that the techniques and the tools that are used for studying those systems are sometimes applicable to brains. So the mathematical tools that we're doing, the modeling, some of the modeling that we're doing really derives from people studying systems of other types. So there is this underlying universality of complex adaptive systems. They all seem to have very similar characteristics, and the concepts, the ideas for studying them all kind of converge onto these mathematical structures, if you like. There's mathematical ways of doing things. So some of the ways that we model how action potentials fire in time are derived from models of other complex systems where people have tried to do the same thing.
Yeah. Yeah, that's really interesting. So your focus is on children and the developing brain. Talk a little bit about that complexity and that adaptation through human life, from the development to aging. What do you know, or what do we know, and how do you think about it?
So I think about that probably in a pretty standard way. So children or babies are born not knowing very much, and the amount of adaptation they do over the first couple of years of life is enormous. So there's a level of self-organization that's happening from a very early time that's fast. And that's babies developing in the first couple of years of life, getting from doing not very much to walking, talking, jumping, all the other things that babies do, which then evolves further into cognitive things. Children are then really learning counting, numbers, all the way through school. All of these things are adaptations. Memories are adaptations. Learning is adaptations. And I think that just continues through life. At some point, the brain is less effective at doing that. Whether exactly that happens is not entirely certain, but it's clear that with aging, there is less adaptability. We are less able to make new memories, less able to learn new things. And in the context of age and disease, even though I'm a child neurologist, I do study a model of Alzheimer's disease. We are interested in how neural networks change their behavior with aging and how the Alzheimer's processes, pathophysiological processes, are disrupting those networks, what those patterns look like.
So tell me a little bit about epilepsy. I have not had anybody in my family or known anybody that went through that with their child. Or what is that, and why does it occur?
So that's a generic overarching diagnosis for people that have had 2 or more unprovoked seizures that are more than 24 hours apart. So by definition, that's what it is. It is a consequence of some underlying brain disease. So I think that's a big part of the way I conceptualize it. At some level, epilepsy, the seizures are sometimes conceptualized as the disease. I tend to conceptualize them as the symptom of some underlying disease. And then when you think about the underlying diseases, they are many. Just about anything that can hurt the brain can set it up in this way that it is able to change into something that looks like a seizure and then stop. So I tend to think about this as brain states, just like the brain is in a state of being awake or asleep or learning. It's constrained. So our brain networks are constrained to only make a certain number of normal behaviors. And when the brain is broken in some way, somehow that constraint is loosened, and loosened in a way that in some people, the consequence is a different brain state, which is the thing we observe and call a seizure. And when we go about trying to treat seizures, we are effectively trying to put that boundary back in. At a conceptual level, I think that's what we're doing. At the level of science, we're usually trying to do something like manipulate a protein, manipulate a receptor. And we draw the straight line. I have difficulty with these straight lines. We say, if you block this sodium channel, the person is less likely to have seizures. And as an observation, that's true. But the sodium channel per se is not responsible for the epileptic seizure. The network is responsible for the epileptic seizure. So we seem to be sometimes too low down in this hierarchy for the way I like to think about these things. So somehow the way that the drug binding at a receptor is changing the way the neurons fire, is changing the self-organization principles that are happening inside the network, somehow reestablishes a boundary. And I don't necessarily need to know exactly how that happens. But if we can achieve that and learn what the neural network behaviors are that constitute the boundary, then that's actually the thing that I want to try and fix. I don't necessarily want to go and do something at a protein. I want the network to be reorganized in a way that it's no longer capable of making the seizure, and hopefully more capable of making cognition.
Yeah, the way you describe it too, it just seems like it's different in every person because we all have a network, but how our individual network adapts may be different than somebody else. Is that true?
To a point. So I think there are things that our networks all do. So if we measure EEG in people, so you put electrodes on their brain and you measure their brain waves and you close your eyes, everybody makes a particular rhythm at the back of their brain with a bit of variance around it, but nevertheless, they all make that type of rhythm. So even though we've got different networks, there is convergence to certain numbers of patterns. And I think this is the thing that's true about complex adaptive systems and makes them tractable at some level, is you can study things that look very complex and very complicated. But if you can identify the overlapping pieces, if you can identify the things that are consistent, then that becomes a target. That becomes a therapeutic target in the same way that gene therapy becomes a therapeutic target in my mind. And this is exactly what we see. So we study complex adaptive systems at the level of single neurons and how they fire in time. So neurons fire action potentials. The action potential is the fundamental unit of information transfer, so like a bit in a computer. So when it's firing action potentials, it's sending information around the brain, and the consequence of that is behavior. The reason I can move my hands and talk to you is because I've got action potentials firing in the right place at the right time in my brain and sending that information to the parts of me that needs to be able to speak. So what we look at is whether there are consistent changes in those patterns, particularly in one part of the brain called the hippocampus, across a range of disorders. And what seems to be emerging from what we're doing is many diseases. You can break the brain in many ways, and the pattern that emerges out the other side is very similar. The abnormal pattern is very similar. So that then raises a very nice hypothesis that if we can encourage the brain to make more normal behaviors, then we might be able to help people across a very broad range of neurological disease. You can then step back a little bit from the idea of, I need to find the gene or I need to find the cause and fix the cause and that's going to fix the thing, is okay as an idea. The problem is there are 1,000 genes associated with bad epilepsy, 2,000 genes associated with autism, multiple other trauma, malformations. There's so many things that underlie seizures and underlie cognitive impairment. If we're going to fix them one at a time, that's going to take us a very long time. And even then, conceptually, that depends on the idea that there's a straight line between fixing the element and getting the outcome that you like. And that may not be true. That's just a generic statement about complex systems. That just may not be true. So I don't have a problem with people doing that. I think people should do that type of research. But I think opening people's eyes to this idea as an alternative approach is also strong. This should also be thought about, and research should be done inside this frame, I think.
So I think you said at the beginning that it's a new way of thinking about, brain research and how the brain's working. Do you find, since it is a kind of a new, and there's probably limited people looking at it this way, do you find support for these, willingly support for these new ideas, or is it more difficult to find people that actually want to kind of invest in this research because it is so new?
The latter, I think. There are very clear, long-standing approaches to science which fall into a biomedical reductionist framework, which is kind of orthogonal to everything that I'm talking about. So the main idea that's out there for understanding brain disease is find the cause, manipulate the cause, the brain will get better, everything will be fine. I see enough evidence of that not working in certain circumstances that I'm not enthusiastic about doing that myself. So in the Alzheimer's world, clearing amyloid has not had a dramatic improvement in people's cognitive abilities. And the idea of precision therapy, the idea that the big idea that's out there is if you can find the cause and fix it, all will be well. And that doesn't seem to be the case. And there's many other examples across neurological disease where very specific things that are supposedly directly related to an outcome can be manipulated and in fact don't generate the right outcome. In some ways, this is an offensive statement to people that are doing that sort of work. And I'm disappointed in that because I don't think I'm trying to be offensive. I'm just observing what I see and stating it. So when I first heard about complex systems, it came from that background of, well, so much of this just doesn't seem to be true. So much of what I'm being told doesn't seem to be true. And the whole complex adaptive system world opened up to me and I was like, wow, now I can grasp ideas. These things fit into my worldview. And I kind of had the sense that other people would also do that. Turns out that that's not necessarily true. So it does feel uphill when I find people who are enthusiastic about it. They're very enthusiastic. I think when you get to traditional funding bodies, then we do start to run into more difficulty, that this gets seen as fishing expeditions, not well grounded in science. What's the mechanism? When people are saying what's the mechanism, they're saying what receptor or what gene. And it requires that whole reconstruction of the whole thing from the ground up to there is never one gene, there is never one receptor, there is never one cause. That idea just doesn't carry any weight in this systems framework. And I think that's a hard idea to put to bed.
Yeah. This, I think it was this week, the Allen Institute, Brain Institute, sent around a paper, and there was a beautiful picture of they have inside the mouse, the network, right? The network inside the brain of the mouse. Maybe those type of pictures and those type of visuals will allow people to understand that complexity and kind of understand that it is more of a systems of systems. And maybe that kind of, evolution will help. That says, hey, we do need to look at this, kind of holistically, if you would. And then there's science is looking at other parts of the body and in its relationship to the brain and maybe, the cardiovascular, the immune system. And, maybe that will, kind of trend this way of it is a system. It is a complex system. So hopefully that'll be the case. So, when you're treating children, because you are a clinician, you are a doctor in there, and somebody brings their child in with the seizures, what kind of hope do you bring them with this idea that, the study, the research side around complex adaptive systems? How is your process with that patient maybe different than others?
So I think at the less acute end, I behave like everybody else. So there is a large number of children who have a few seizures. You give them an anti-epileptic medication. It's effective. The seizures go away. And over a couple of years, their brain matures in a way that means they no longer have seizures. They come off the drug and they're fine. Those are not the type of children that I'm really trying to pursue here. So that's 70-ish% of children will have that reasonably well-controlled epilepsy. There's another 30% that are much more problematic. So the other 30% are the children that have epilepsy, and then they are often associated with other things. They have cognitive impairments, memory impairments, low IQs, struggling to attain things at school. They often have autism spectrum disorders, ADHD. So we've shown, amongst others, epidemiologically, that's a very heavy tail in epilepsy. There are many, many more children than you would expect with those sets of disorders. The way that idea has been constructed for a very long time is as an epileptic encephalopathy, is how it's described. So the idea there is that the seizure per se is causing harm to the brain in some way. And because of the seizure, you're getting the epilepsy, you're getting the autism, and you're getting the cognitive impairment. This is a very attractive idea that really captured the imagination of people in the epilepsy field. And it's attractive because it says if you fix the seizure, you fix everything. If one's causing the other, then fix the seizure and magic will happen. When I first started, 25 years ago, I guess, I started seeing these children at Great Ormond Street, where I was practicing at the time, and I just couldn't see it. We were making these children better. We were reducing the numbers of seizures, and their cognition and their autism just never got any better. And at the time, what we were doing was chasing epileptic seizures very heavily and chasing abnormalities in the EEG. So these children were getting onto more and more and more anti-epileptic medication because if we can fix the seizure, we'll fix everything. So I started doing the exact opposite. I started reducing medications in these children. And I would say to parents, your child's got the worst of all worlds here. They've got a cognitive impairment. They've got autism. They've got seizures. And the seizures are not getting better. And now we're making everything worse because we're giving you drugs with lots of side effects. And we're giving you lots of drugs with lots of side effects. And in fact, what I have done consistently over a long time is try to have children on 1 or 2 medications at standard doses, accepting the seizure, saying your child's going to have seizures. That's the way of it. But that's not what it's about. If your child has 5 minutes of seizure a day, which would be dreadful epilepsy, then they have 23 hours and 55 minutes of no seizure a day. And I don't want to disrupt 23 hours and 55 minutes in order to somehow minimize 5 minutes. That doesn't make any sense. And parents understand that. So they are happy to wind back the medication. And what I see is a lot of children who are more alert, more interactive, more able to access school, more able to access extracurricular activities. All of those sorts of things happen because I minimize the impact of the drug, which is fine, but it's kind of small. I have more ambition than that. What I want to do is to recover the cognition and the behavioral things, the autism. I want to improve those aspects. So while we live in this world of the seizure causes stuff, and the most important thing is to treat the seizure, I don't know that we're going to make much progress. So my approach, both clinically and from a research perspective, is really to say the inverse, is to say the brain that's got a broken network and is able to make seizures, is able to make this new state, is also able to do these other things. It's also a brain that is unable to compute and make cognition. It's also a brain that's unable to allow you to be sociable and not make repetitive behaviors as in autism. So in order to fix all of those things, if all of those things arise from an abnormal network, then what I need to do is fix the network. I need to fix the underlying thing. And that then becomes an entire research program of how do you go about doing that. You need to understand the network. You need to start to understand ways to intervene inside networks, observe whether you can recover networks, and observe whether you can recover cognition and behavior. And I've done that really over time, moving more and more and more away from epilepsy models to trying to understand cognition directly in an Alzheimer's model or in a Fragile X model of autism to look at behavior. So I'm now much more interested in trying to understand those networks, because I think those are the same networks that exist in people with epilepsy. And my intuition, rightly or wrongly, is if I can recover the cognition and the behavior network, the consequence of that will be a network that's not able to make seizures. Have I shown that? No, not yet. But that's the line that we are going down.
So what does that mean inside your lab then? Like, give me some idea of what that portfolio of effort, your experiments or your studies to study the network. Because I would imagine that you'd have to break it down into pieces and parts and build it back up, as far as your effort, not necessarily the network itself. Do you use mouse models? Do you use human tissue? How do you do this?
So at the moment in the lab, we use models. So I used to use rat models quite a lot. We've now moved more to using mouse models. And I'm at the level of single unit electrophysiology. So the thing that the basis of our work is we implant electrodes into the part of the brain called the hippocampus, and sometimes the hippocampus and the prefrontal cortex. So these are subsections of the brain that are really underlying a lot of the memory, behavior type things that I've been talking about. And what we do is we implant electrodes. We're able to then record multiple neurons, which we can isolate as single neurons, in time while animals are doing things. So they're awake, alert, they're doing whatever we want them to do. And then the downstream of that is we capture all of this information, which in the end is zeros and ones. If the brain is not firing an action potential in time, it's a zero. When it fires an action potential, you get a one. So now all we have is a time series of zeros and ones that we want to model and try and understand the patterns that exist inside that binary data. So that's where all the math then comes in. So the math and the coding, so we have mathematicians in the lab as well as biologists, are then coding in order to say these are the patterns. And we've seen some very characteristic patterns. So when a hippocampus is making behavior, it doesn't just have a string of action potentials. It has bursts of action potentials that burst at a certain frequency. And if that bursting pattern is altered, then when you look at those animals and evaluate how good they are in a task, they are not good in the task. So we've shown that across the hippocampus and the prefrontal cortex and how those two brain regions are talking to each other, and shown that at all levels, that if those patterns are disrupted, whether it's one neuron at a time, multiple neurons, how neurons talk to each other, correlations, all those sort of mathematical things, they all predict that animals are going to be less smart in whatever task you then put them in. a very interesting experiment that we did, which I don't think captured people's imagination, is we took animals that had a malformation of cortical development. So their brains were malformed. We gave the mothers a drug, which meant that the babies all had malformed brains, and the animals had a cognitive impairment. What we did is we simply took those animals and either grew them up with friends and toys, environmental enrichment, or we left them in standard laboratory conditions. And the first thing, which maybe is not that remarkable, the animals that were environmentally enriched were much better at solving a maze than the animals that were not environmentally enriched. That's kind of expected. What was remarkable is their neural networks recovered. So the animals that were environmentally enriched had much more precise, high-fidelity burst firing going on in the hippocampus. It was much more normal firing than we saw in the animals that grew up in standard lab conditions. So the inputs, we changed the inputs into that system. That's all we did. We just environmentally enriched. So they got different sensory input through a whole range of different means. And that sensory input meant that the brain was able to reorganize and self-organize in a way that it made patterns that were much better and much more normal and were associated with improved cognitive behavior. I still think that's an amazing finding. It doesn't take you very far because environmental enrichment is a bit blunt. But where it's taken us is to brain stimulation. So what we are now trying to do is we put electrodes into the parts of the brain that project to the hippocampus and the prefrontal cortex, give electrical impulses at different frequencies at different times. We explore that space in order to see if we can drive the network to make more normal behaviors and whether that then leads to improvements in the animal solving the mazes. And we have some remarkable preliminary data that isn't published yet, which is showing exactly that in a Fragile X model, if we stimulate at the right frequency, in the right place, the animals become more sociable, they're less anxious, they're more able to solve memory tasks. And in fact, their network, not in vivo, so the ex vivo network, if we take the brain from the animal and record electrical activity in those that were stimulated and those that weren't, the animals that were stimulated have more normal hippocampal networks at that level. So this is all just driving me to, we need to find the right places and the right frequencies and the right types of inputs into neural systems, even if they're broken, because the network wants to be better. The broken network wants to make cognition. It wants to make behaviors. And if we can just gently push it, give it a bump, it means that it organizes in a way that it can do it, then that seems to me like it's going to have an enormous impact. And if we can do it across disease, if all of these models show similar things and we can show similar bumps lead to improved outcomes, then at some level we can start to move away from the idea of I need to find 2,000 different treatments for autism to brain stimulation is effective across many neurological diseases. How do we go about doing that in a clinical way? So it comes from the patient, I think you've got a broken neural network, to modeling it in the mouse and modeling it across different mice and then trying to fix the network. And now starting to show, we're getting down the other end of showing that we can fix the network with brain stimulation. Does that then ultimately end up back at, brain stimulation for people with epilepsy or autism or Alzheimer's disease?
So there are kind of emerging technologies out there that are trying to stimulate parts of the brain for depression and is this what you're kind of leading towards? Is that maybe there's more advancement in that area where people could either go to a clinic or, wear a cap a certain part of the day and be able to, like you said, repair the network or whatever it is that may be malfunctioning?
So yes, I think this is the blue sky end of this. So where brain stimulation is very effective is Parkinson's disease for deep brain stimulation. Transcranial magnetic stimulation for depression is effective. We're starting to do some TMS work in autism to see whether we can improve outcomes in children with autism with TMS. The thing that is universally true, I think, certainly mostly true, is when we are designing the stimulation paradigms in all of those circumstances, we're kind of hazarding a guess. And we don't really have good markers of this is the right frequency. So the thing that I was saying about the Fragile X model is what's interesting is you have to stimulate at the right frequency. We tested multiple frequencies and only one of them gave us an effect and gave us an effect multiple times. So I don't think it was a fluke. I think that that's genuinely a frequency that those brains like to see in order to improve outcomes. We don't do very much of that exploration of parameter space or looking for markers that say, this is what the abnormal network looks like in the human. How do we recover that? Can we measure it inside the brain? We are much more, let's try this and see. And in epilepsy, that can be quite hard because children have fluctuating epilepsy frequencies. So you have to do it over time. It takes a long time to tune it. So I think where we are going to see progress, or where progress probably needs to be made, is in that tuning component. We need to know where to put electrodes. We can do that robustly. People are very good at placing electrodes into very small pieces of brain. We then stimulate at, let's try that. That didn't work. Let's try this. That didn't work. I think if we can now take the jump to more precision type things, can I, in some way, buzz the brain and observe an outcome in that network? Can I show that the network is now behaving more normally or not? And if it's not, buzz it again in a different way and somehow explore through that area the right frequencies, the right stimulation paradigms. Maybe that then is the next frontier. And that's what we're trying to do in the animal. We're trying to show that if I stimulate in this spot at this frequency, am I actually making this network behave more normally? And can I measure that in real time effectively? I think that's the direction I would like us to be going.
So you've published over 140 papers. I think you've already answered this question, but what is, maybe the most impactful finding that or conclusion that through those papers that has really kind of driven your research?
So I think there's different parts of this. I've done very broad-ranging research from drug trials through epidemiology, through human cohort trials, animal studies. You do a lot of MRI, a lot of EEG. And the thing that's probably had the biggest impact is something we've not talked about at all, which is a rescue treatment for children that have long seizures. So if children are having seizures that last more than 5 minutes, those seizures are likely to continue for 30 minutes or more if you don't intervene. Before I started, so in the early, probably from the 70s to the 90s, the treatment for that was to put a medication into the rectum, which comes with its social concerns and practical difficulties. So one of the first things I ever did was test whether we could give a very similar type of drug, just pop it into the side of the mouth and see whether that worked. And it works very well. And that is now pretty much a standard of care across Europe, Australia, New Zealand, probably much of Asia. In the United States, they went they really went for the nasal approach rather than the buccal approach. And that's now much more common in the United States. So in terms of what thing did I do that's had the biggest impact across a whole bunch of children, that very simple idea is the one.
Wow. I can't imagine being a parent going through that for 30 minutes or so with your child. It has to be scary. It has to be just horrible. Like, that's amazing that you did that. What do you think, with people that are kind of out there, what are some of the misconceptions about epilepsy that you think, you'd like to kind of overcome or that you address with your patients?
I think the biggest misconception is that seizures are harmful. And that's really the one that I would desperately like to see go away. I can persuade parents of it, but it's sometimes tough. So when children first have their seizure and 911 is called, or the child is coming to an emergency room, the thing that happens is everybody runs around. Everybody is putting oxygen on the child, trying to get blood, giving drugs. All sorts of things are going on that look very, very scary. And the consequence of that is the parents think that their child has got a very nasty thing that I then have to backtrack on. It's really difficult. At the outset, it can be difficult to persuade parents that, in fact, all of that was kind of unnecessary. Didn't need to be quite that fast, quite that aggressive, quite that scary. We could do this much more slowly, much more thoughtfully. So I'd love to be able to persuade emergency rooms to be gentle. It's okay. The child's having a seizure. Give them a drug. Wait. Be reassuring to the parent. If you can do all of that stuff up front, then it's much easier downstream to say seizures are not harmful. It's okay. What we need to do is think about quality of life. So for me, that's the biggest misconception. I do believe that that's moving, that there are more and more people that accept that the seizures are not quite as harmful as they thought they were. I think I'm still in a minority that thinks they're largely not harmful at all. But yeah, there's more and more acceptance of that view that you don't have to treat every seizure, you don't have to treat every spike on an EEG. It doesn't necessarily improve outcomes. So I think that's the one. And the other one that, again, is a simple idea, is that anti-epileptic drugs have side effects. And it's probably true across a lot of medicine. We give drugs and go, it's going to make you better, and really don't have enough consideration of what those medications are doing. And in the epilepsy field, there are statements like, our goal is no seizures, no side effects. So how do you do that? So what you're suggesting is you're going to give a drug to a child that's going to go and live in the brain. It's going to alter the brain because it has to in order to stop seizures. And the only consequence of that is no seizures. Surely there's a consequence in cognition or sleepiness or behavior or something. And that's exactly what you see. The children always have some level of side effect. And that's what I was saying at the beginning of the session, really, was we need to withdraw some of these medications. We need to understand what we're doing. So a greater understanding of the negative impacts of medications and a greater understanding of the limited impact of epileptic seizures are the two things that I'd really like to see changed in the epilepsy field.
So you've worked in the UK, you're from Africa, I believe, South Africa. So you've worked there in the United States. How are these, I would imagine there's commonality with how we think about things, or there's probably some different ideas. What are those kind of differences that you have seen?
So I trained in Zimbabwe. I think the thing that I learned in Zimbabwe was reliance on clinical skills. So in that environment, you just have to make decisions on the basis of what you've heard, what you see, physical exam, without access to intensive resource things that are available in developed countries such as the UK and the US. I think what that taught me was the ability to see a sick child, to understand a sick child compared to a not sick child in this morass of children. We're seeing so many people. There's so few doctors. So you really have to be able to pick out the one that's the sick one in the crowd. So I think I got very good at that and very good at depending on my clinical skills. That did carry over into the UK. So obviously I learned much more nuance. It's where I became a neurologist. It's where I became a neuroscientist. I had to learn much more nuance about things. But even now, I think I use some of the tools less frequently than some of my colleagues. Some of the technology, I don't just screen. I tend to try and use it because there's a question that I'm trying to answer. And then between the US and the UK, it's very similar in the end. I mean, they're different systems. The UK has a universal healthcare system, and that has its advantages and disadvantages. And the US does not have universal healthcare, which comes with its advantages and disadvantages. But at the level of medicine, at the level of how I think about the children, I think it's the same. It's the same across all of those environments. In the end, it's about the brain.
Tell me a little bit about Namirs and the Children's Hospital and where you are right now, and put a little plug into what makes it special.
So Nemours Children's Health is a pediatric healthcare system that's multi-state. So I'm at Nemours Children's Health in Delaware. So I'm in Wilmington, Delaware. And as division chief here, there are 15 neurologists that have a range of subspecialty interest. So we have several epileptologists, but a neuro-ophthalmologist, headache specialists, sleep specialists, neuromuscular program. So we are providing subspecialty neurological care to a broad range of children. And in the epilepsy group, which is obviously the one that I have the most interaction with, we are doing a lot of the things that I'm talking about. I do drive these ideas of less medicine, less scary. So there are research projects going on within the hospital, which is really trying to achieve that sort of thing. So at some level, I think that's kind of what makes us different. We have a different construct for some of the ways that we are dealing with these diseases. And from my experience, the parents like it. The parents like to come to us and experience the slightly different, particularly those that have gone through the mill and gone through the very standard, many things have been done to the child that haven't worked, to come to an environment where we go, yeah, it's okay. Let's help you maximize quality of life. Let me take away some of your fear about what these things are. I think makes a big difference. So Nemours is also in Florida. So there are at least 2 sites. In fact, there's 3 sites, biggish sites in Florida. And the goal here for me now is to try and export some of those ideas throughout the enterprise and see whether we can start to grow this sense of there are alternative ways of thinking about epilepsy to the ones that are standard.
So how are you going to do that? I mean, through the researchers or communication campaign, or how?
Partly through the researchers and partly through the clinicians. So I think the teaching of residents has become critically important. So we have residents for 3 years in pediatric neurology, and they stay in the same environment for that period of time. So I think because we are now becoming much more coherent in the way we think about epilepsies, what our residents are seeing is 3 years of a style that's not necessarily a standard style. And they're not going to experience much of the other style because they're not in the other hospitals. They then go out and become attendings and do other stuff. And I think that's happening within our residency, but the other Florida sites also have residencies. So there's a level of teaching that needs to be done there, a level of changing the culture. As we do that, I think it will expand. In order to convince people, we need the research. In order to convince people that it's true, I need to be able to say, here's the data, here's the evidence, these experiments were constructed to answer that very precise question, and try and give them some reassurance that I'm not promoting something that's dangerous and harmful.
What are the questions or the top questions that other researchers that, through other universities or such that are working in this area of childhood brain development or epilepsy or autism? What are a couple of the top questions you think that need to be answered by the research community?
Generically or by other people. So I think the questions that I think need answering are the ones I've talked about, which are the neural network things. I would say there's way, way more work that's being done at the level of genetics in epilepsy and autism or behavior. So certainly in the autism world, the use of ABA as a way of modifying behaviors in children with autism is certainly gaining a lot of attention and worthy attention. I think that it really is having positive impacts. Yeah, the bit that I struggle with is the one gene, one disease. Let's work out some very expensive technology to put a gene inside a brain, and this is going to have a positive impact. I don't really buy into the view that we should keep finding genes. So the catalog is now very big. When we had a catalog of 5, that seemed okay. I'd much rather, from the genetic end now, that people choose what they think is the high impact gene and attempt at some way to modify an outcome and show me that in principle, this idea is a good idea. It's being done. There is some gene therapy work being done in Dravet syndrome. It's early days. They are having some impact on seizures. The impact on cognition so far has been unimpressive, but I'm willing to sit back and wait. If that's the bump that works, then that's great. I'm all for it. So again, my construction is not you've replaced the sodium channel magic. My construction is you've done something to the brain. You've changed its inputs. You've changed it, the way it's going to be able to behave, and you've allowed it to renormalize. Do I think that in the future there's both possibilities? I think yes. I do quite like the idea of we're manipulating the cause and at the same time we're driving the brain to try and normalize. And whether that interaction of I fixed the gene and I changed the inputs so that it's self-organized in a better way, is it that combination of things that's ultimately going to be the golden bullet? Maybe. I think that's definitely something we will go to in the end.
So on that note, looking out the next 5 or 10 years, what do you see? Where do you think we'll be, or what are you the most hopeful for in this area of childhood epilepsy, autism, cognitive challenges?
So it's a reasonably short timescale. I think what I'm most hopeful for over that timescale is that we will be better at brain stimulation. So there is an increasing interest in brain stimulation in people with epilepsy, and children with these severe epilepsies are increasingly getting stimulation in various parts of the brain, particularly in the thalamus. And I'm excited to see what that brings. So at Great Ormond Street, I still do a bit of work in the UK. They are doing work on thalamic stimulation, as is other people in the world. And I'm hoping that I'm going to get my hands on some of that EEG data to see if I can explore the network consequences of what they're doing, where those studies are really set up to see whether we can treat epileptic seizures. But that link between here's somebody who's interested in networks and here's somebody who's interested in stopping epileptic seizures, and we have something in the middle that both of us want, seems like an interesting interaction that could take us forward.
Well, this has been fantastic. I'm taking up your time, and I'm sure you're very busy. As we wrap up here, how do people get a hold of you? Whether or not they're possible patients or researchers that want to talk to you about this complex, adaptive network theory.
So I can be found on LinkedIn. Unfortunately, I don't keep that very up to date. So you can access me through there, but it probably doesn't carry much information about me. You can look at the Nemours website. Nemours Children's Health website will have me and what neurology is doing and what neuroscience looks like at Nemours. And then I can be contacted by email, which is rodney.scott@nemours.org. And I'm always excited to talk to people about these ideas. I'm always excited to see how we can move this forward.
Thank you so much for your time. This has been amazing, and I will give you the last word.
Great. Well, thank you very much. I very much enjoyed doing it, and I hope I've gone some way to trying to change some people's minds.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.